Characteristics of the genetic markers. What are genetic markers? Any phenotypic difference controlled by genes that can be used for studying the recombination processes. They are called as genetic markers. There are different characteristics of the genetic markers. Here we can see that what are the common characteristics of genetic markers. If we see about the number of loci, in case of morphological markers, protein markers, RFLP markers, RAPD markers, and SSR markers. In case of number of loci, they are limited, almost unlimited, but in case of RAPD, unlimited, and SSR markers, they are high in case of number of loci. While in case of inheritance, morphological markers, they are dominant, protein markers, they are codominant, RFLP also codominant. Inheritance in case of RAPD markers is usually dominant, while in case of SSR markers, that is codominant. So these are some of the general characteristics of the genetic markers. Other characteristics of the genetic markers in the form of positive features and in the form of negative features. In case of morphological markers, protein marker, RFLP, RAPD, and SSR. So positive features, they are that morphological markers, they are visible, protein markers easy to detect, RFLP utilized before latest technologies. In most of the cases now they are not used. Quick assay with many markers, well distributed within the genome. In case of SSR markers, they are well distributed within the entire genomes. When we look at the negative features, that possibly negative linkage to other characters, possibly tissue specific in case of protein markers, radioactivity required rather expensive RFLP, and RAPD marker have high basic investment. In case of SSR, long development of the marker, they usually are also considered as a little bit expensive. What are the characteristics of the molecular markers? So molecular markers, they can be in the form of RFLP, DFP, RAPD, SSR, and SNP. So the detection method is hybridization. In case of DFP is also hybridization. In case of RAPD, SSR, SNP, it is through the polymerase chain reaction, which is also called as PCR. Type of the probe or the primer used, and that can be a genomic DNA and cDNA in case of RFLP. In case of DFP, mini satellites. In case of RAPD, they are arbitrarily designed primers. Sequence specific in case of SSR and SNP, both are sequence specific primers or the probes. And need of activity in almost all. Initially, need of the radioactive were used, but now in most of the cases, there is no need of the radioactivity. Characteristics of the molecular markers in the form of extent of genomic coverage, they are limited, extensive, and all these are DFP, RAPD, microsatellite, and SNP, they are extensive and found throughout the genome. Degree of polymorphism in case of RFLP, it is low. In case of DFP is high, RAPD medium to high, and microsatellite and SNP polymorphism is also high. Phenotypic expression is codominant. In case of DFP is also codominant, and microsatellites, they are usually dominant, and SNP codominant. Possibility of automation in case of RFLP and DFP is no. But in case of RAPD, microsatellite, and SNP, there is possibility of the automation. There are many different types of the genetic markers which are used for the calculation of the recombination frequencies, or they are used for the genetic mapping of the genomes. If we conclude what is genetic markers, any phenotypic difference controlled by genes that can be used for studying the recombination processes are called as genetic markers.